Okay, I'm sure you're sick of me now, but this is what we got to do. This is what we got to do. Um, we just finished talking about independent assortment and segregation of chromosomes during the process of gamete formation, and realized that really when we're talking about genetics inheritance, we're really just talking about the possible ways that alleles can be rearranged and separated during the process of gamete formation. And then again, it's a crapshoot as to whether which gamete fertilizes the egg and passes on the genetic information of that organism. So, a few probabilities stuffs here. Let's go over here. One parent has a genotype big A, and we don't have to give this what the A's names are, what character is. What is the probability that one parent's offspring is going to inherit the dominant allele here? What is that probability? And the second part, what is the probability of one parent's offspring inheriting the recessive allele? All right, so if you said the probability that one parent's offspring inheriting the dominant allele is 100%, then you would be correct. The probability of parents inheriting the recessive allele, part B, is 0%. And if you have any questions about that, please write them down. We'll talk about them in class. Second part, one parent has the genotype big A, little a. What is the probability of one parent's offspring inheriting the dominant allele? Second off, what is the probability of one parent's offspring inheriting the recessive allele? All right, for part A, if for part A you said the probability is 50%, you would be correct. If for part B the probability is 50%, you would also be correct. All right. Cool. All right, let's move on a little bit more. We talked about Mendel's monohybrid cross, so we don't have to talk about that anymore. The way you're setting up a Punnett square is just on one side you put the possible allele combination of one parent with the possible allele combination of another parent. Now if you look here, you have the frequency of, or the probability that this organism would pass on one or the other allele. Remember, at the metaphase plate, you have one chromosome with the big B on it and one chromosome with a little b. Now, what are the chances of one going one way and one way another? Well, the chances of big B going to the left and little b going to the right, all right, are 100%. But what are the chances that this gamete fertilized the egg versus the chances that this gamete fertilized the egg, or that the egg, the gamete that holds the little b fertilized the egg versus the gamete that holds the big B fertilized the egg? Well, it's 1/2. So I just put a 1/2 here. The chances that this organism donates this big B is half, and the chances of donating a small b is another half, equaling 100% chance that either one is going to be inherited. Now, if we go to the other parent on the other side, it gets the same thing. All right, the chances that one of the organisms will have a pollen grain that has the big B is half and the small b is half. Now, all we do, all right, to find out the frequency of big B, we multiply, all right, the frequencies of each single event. So the frequency of donating this B is a half, and the frequency of donating this B is a half, so the frequency of the offspring being this is a quarter. All right? And do the same thing all the way around. Now, one of the questions is, what is the probability that the offspring, all right, are going to inherit an allele combination that makes them display the dominant phenotype, or purple? Right? In this case, you're going to add, all right, the probabilities together. 1/4 plus 1/4 plus 1/4 is 3/4. So the chances that the offspring inherit the dominant phenotype in this case is 75%. All right. If we said, what are the chances that the offspring are going to inherit either a homozygous dominant phenotype or a homozygous recessive phenotype? Again, when you hear or, you're going to add the two together. The chances that we get this or this is going to be 1/2, 1/4 plus 1/4. All right. Now, here's the tough one. What are the chances that the first offspring is going to be homozygous recessive, and the chances that the second offspring is going to be homozygous recessive? Well, now we treat these as two mutually exclusive events, and when we multiply them together. All right. So the chances of getting an offspring that's homozygous recessive is a fourth. All right. Now the chance of it happening again are no different. They're a fourth. So the chance of them happening together, all right, one after the other, is a multiplication of the two. So the answer is basically 1/16. All right. So that's the rule of addition, all right, or the rule of multiplication. The rule of addition says, what are the chances that I get big B, little b? The chances are half, because chances of me getting it this way are a fourth. Chances of me getting it this way are a fourth. The chances of me getting it at all are the addition of the two, all right, which is a half. Okay, so these are some probability problems. I want you to work through these on your own. We will go over these in class. Please remind me. I might even assign them as homework. I'm not sure. This is part 4, so it's a few days away. So if you can answer all these questions correctly, or at least answer them wrongly, and then we'll go over them in class, and you'll have a good idea about why you might have answered them wrong or right. Then you have a really good hold on how the laws of probability, multiplication, and addition help us to solve genetics problems about probabilities. Because remember, generally things aren't so easy. You're more likely to be solving a genetics problem like this with 3 different characters, or maybe even 10 characters, than you are with 1 character. Okay. Now, test cross. All right, what is a test cross? A test cross is designed to find out what the genotype of an organism is that is displaying the dominant phenotype. Because remember, an organism that is displaying the dominant phenotype, remember, we go dominant phenotype, could either be heterozygous or homozygous, all right? Because the heterozygote is masking the recessive allele, displaying the dominant phenotype, and the homozygote also has a dominant phenotype, but might have, 2 dominant alleles. So a test cross is designed to figure out what your genotype is of an individual who's displaying the dominant phenotype. Now, if we take the dominant phenotype individual, which we don't know what they are, big P, or big P, small p, and we cross them with a recessive phenotype of the unknown genotype, all right, small p, if we get any offspring, all right, that are displaying the recessive phenotype, and we know that this organism must have the possibility of donating their recessive allele. If all the offspring are all the dominant phenotype, all right, then we know that this organism can only donate the dominant allele, which will be leading to all heterozygotes. All right. So remember, a test cross is important because there are many times where we have a dominant phenotype of a character that we want to select or we want to artificially breed, but we want to know, we don't want to breed that organism if it's a heterozygote, or we don't want to introduce, the undesirable allele into the population that we're trying to do. All right. So there's a few other patterns of inheritance here. One of them is called codominance, where neither allele is dominant over the other. All right, this is neither allele is dominant over the other. And in examples of codominance, when both alleles are present, all right, the heterozygote shows the phenotype from both alleles. All right, we're back online here. Just displaying, so before we had things like big T, small t, big S, small s, big P, small p. And this is only when one allele is more dominant than the other. All right. Now, codominance is a little bit differently, that both alleles display their phenotype when in the heterozygote. For example, we take a true breeding, white-colored car. Car was stupid. I apologize. We take a homozygous, true breeding, white-colored plant, a white-flowered plant. All right, color. C means color, and W is the allele. So color is the character, and this is the allele. Color is the character, and now we're talking about red here is the allele. All right. and we cross them, all right, this organism can only donate the CW, and this organism can only donate the CR. So we end up with the heterozygote. Now, in examples of codominance, this organism is both white and red, just like in the flower shown here. All right. So in examples of codominance, the only difference is the heterozygotes are showing both phenotypes. All right. Neither phenotype is being masked. All right. So that's kind of important that we understand that. And also, we write the alleles differently in codominance. Now, we also have something called incomplete dominance or intermediate inheritance. All right. Again, let's use the example of flower color. We have a true breeding, red-colored flower plant that is being crossed with a true breeding, white-colored flower plant, and we all know that this organism can only donate one. We can only donate one, so now we have our offspring, our heterozygote, all right, which is no different from codominance, except in intermediate inheritance or incomplete dominance, this offspring is going to be an intermediate, like pink. So both of the alleles are playing a role in the resulting generation or the resulting phenotype, but neither one of them is dominant. They are mixing. They are blending one another. All right. Man, are we cruising for a bruising right now? So we just talked about codominance and intermediate inheritance or incomplete dominance. Our next example of hereditary differences in alleles and things is called sex-linked genes. All right. Now, if you remember, we have sex-linked genes. Let's call it. Look at a karyotype. I was looking at some news on Ukraine stuff. A karyotype videos. I like the colored ones here. Oh, let's look at this one. Okay, we bring it back down. I didn't mean down that way. What you notice here about the karyotype is chromosome 1, chromosome 2, chromosome 3. All of these things are homologous. The only one that is not homologous is the X and the Y chromosome. Look at the Y chromosome. It's so freaking small. All right? What that shows, males have an X and a Y. Females have two X's. What really shows you that the X chromosome is way more functional than the Y chromosome. The Y chromosome only has stuff that is important to maleness, but the X chromosome has stuff that's important to maleness and femaleness and in general being a person. All right? So there's a lot more stuff on the X chromosome than the Y chromosome. The Y chromosome is actually getting smaller, which is crazy. So the question is, what if there are genes that are located on the X chromosome that are not located on the Y chromosome? Well, that would mean that the inheritance patterns would be different in males and females. Well, this dude, Thomas Hunt Morgan, was doing an experiment on fruit flies and eye color, and basically he found a mutant male that had white eyes, a white-eyed male he found. And he also knew that he had something called the wild-type females. And he had true breeding wild-type females, which means that the females they all had red eyes, and they only had red eyes. And then he had a couple males that only had white eyes for some reason. These were mutants. All right. Now the plus, the W plus, the plus means wild type, means they have it. All right, and the W minus means they don't. All right. But if you notice here, the males, since they have one X chromosome, all right, have a trait or have an allele for white eyes because they still have one X chromosome. The females over here in another example, all right, have red eyes. Now, what I want you to focus on now is not the chromosomes themselves, but just the crossing. We take a female with red eyes, we mix it with a male, and we mate a male with white eyes, and what we get is all red-eyed males and red-eyed females. All right. If we take all these red-eyed males and all these red-eyed females and we mate them, what we get is basically red-eyed females, red-eyed males, and white-eyed males. All right? Now here's the deal. All the males here are inheriting an X chromosome from the females. All right? Any male is going to inherit an X chromosome from a female. If this gene for eye color is on the X chromosome, that means this male already having this Y is going to inherit the red eye color allele from the female no matter what, which gives us the red-eyed male, all right, here, which only has one allele because they only have one X chromosome, and the female, which now the female can only inherit the X chromosome from the male, so the females are only going to inherit the white-eyed allele from the males. So that means the females are always going to have, in this example, the females are always going to be heterozygotes in the F1 because they can only inherit the white-eyed allele from the male. And this is often why certain disorders that are occurring on the Y chromosome are only inherited in males, and certain disorders that are on the X chromosome often are inherited from either the mother or the father into the females because they're inheriting only the X chromosome from one another. All right. Now, the important thing about this study was in the F2 generation, only the males had white eyes. All right, only the males had white eyes. And why is that? Well, the males only need to inherit one Y chromosome. All right, or they only need to inherit one white-eyed allele. All right. The females, on the hand, they can be, this example right here, they can be heterozygotes and still display red eyes because the white eyes is actually more dominant over, or the red eyes is more dominant over the white eyes. So the females would actually have to inherit they would have to have a rare white-eyed allele to begin with, and they would have to mate with a white-eyed male as well. Okay, and even then, it's possible that the female could inherit one of the alleles for red eye color, and then it wouldn't matter what she inherited from the male. So the important thing is this, that certain genes are actually linked or tied to specific chromosomes, and especially with the sex chromosomes, males and females are not getting the equal number and equal amount and the same heredity of each chromosome. So this is an example of a sex-linked gene, the eye color. All right. So Thomas Hunt Morgan found that there are certain genes that are located on sex chromosomes, and as a result, that these characters are being inherited differently in males and females due to males and females having different combinations of the X and Y chromosomes. Moving right along here. All right. Sorry, I didn't even press play. I might have. Let's see. Did I press play? I did press play, so I'm good. All right. Sex-linked genes. Again, Thomas Hunt Morgan displaying how the inheritance of a mutant allele, all right, the white eyes, is inherited differently from male to female. All right. So all the females are going to be inheriting this white-eyed allele from the male because it's the only one they get, all right? Which means that only, and because the male is only inheriting the red-eyed, that means only the females are always going to inherit a dominant allele in the red eyes, so all the females are actually going to be red eyes. And then we have half of the males are going to be receiving this allele, okay, from the mother, and half of them will be receiving that allele from the mother, and all the males will inherit the blue here. Okay. Now, something called polygenic inheritance. Polygenic inheritance, where we have multiple alleles that are controlling a specific character. An example here is skin color, and actually eye color is actually an example as well. The phenotype depends on how many and type of allele sets are inherited. So for people that have, so you're going to inherit, there's at least 3 alleles for the that you're inheriting from each parent. There's 6 alleles total that are being inherited separately. So you inherit 3 color genes from the male, from your father, and 3 colored genes from the father, all right? And depending on how many colored genes you have, your skin is going to be different. The albinos are going to be inheriting no allele, or they're going to be inheriting basically alleles that have no pigmentation to them, so they're going to have completely no pigmentation at all. Whereas some people are going to inherit 3 dark alleles from the male and 3 dark from the female, and they're going to end up with very dark skin, all right, because they're inheriting all genes that code for darker pigmentation in skin, all right, and then everybody else is everywhere in between. That's why we all have different skin colors, all right. So this is called polygenic inheritance. There's many genes involved in the inheritance of a specific character, as in skin color. And the last thing we'll talk is about pedigree charts. Now, pedigree charts are a way to determine how specific characters are being inherited within a population of individuals. And we can also tell by the presence or absence of specific alleles of a character in ancestry what kind of pattern that character is likely to follow. Is it incomplete dominance? Is it intermediate inheritance? Is it straight dominant recessive? Is it sex linked? All right. Is it sex linked on a Y chromosome? Is it sex linked on an X chromosome? All right. These are all things that are important. If something is sex linked on an X chromosome, then that means all the females will inherit it from their father. All right. And that all the males, if the mother has it, will inherit it from the female. A 50% chance. If things are only located on the Y chromosome, all right, then only the males, all right, will be receiving the allele because females don't have a Y chromosome. And we will be able to see by this pedigree chart. So in a pedigree chart, the circles are females and the squares are males. The shaded in portions are people that are displaying the phenotype, all right, displaying the phenotype that we're looking at. And the white, the unshaded portions, are people in the ancestry line that are not displaying the phenotype that we're looking at. So for example, if we're looking at phenotype black hair, then all the people in black here, all right, have black hair. And if we're saying black hair versus white hair, then all these people have white hair, all these people have black hair. So the pedigree charts, you're focusing on a specific trait, and if the person shows that trait, then they're shaded in. Okay, that's important to know. Usually, if a person is a carrier, all right, which means a person has the allele but is not showing the phenotype, oftentimes you'll see a half-shaded portion for people that are carriers. We'll talk about more pedigree charts. We'll talk about pedigree charts. Yeah, this is really the end of the Mendelian Genetics stuff. Hopefully, these 4 lectures, plus the information in your book added to our class activity and stuff will be enough for you guys to really hammer down and understand these principles. We will be finishing genetics the week we get back from break, and we will be, really going headlong into one of my favorite subjects, which is evolutionary biology and kind of how genetics works in the ecosystems. So really excited to continue moving on here. You guys done a really great job so far, and we're definitely, we're definitely ahead of the schedule, but we do want to cover as much as possible, so we're going to kind of keep moving along. So I appreciate everything. You guys are great, and if you have any questions or concerns or criticisms, I'd actually love to hear them in a timely manner. And have a great break. Thank you.